that same picture and we add in some of the transcellular components, okay, we can see that we have, first off, ATP pumps, right? Sodium-potassium pumps. And they are arranged the way sodium-potassium pumps usually are. Sodium is getting pumped out, potassium is getting pumped in, okay? Now, that as long as those sodium-potassium pumps are working well, which they are, remember that's one of the main consumers of oxygen in the kidney is the sodium-potassium pumps, right? So as long as these guys are working vigorously, the concentration of sodium inside the cell should be kept relatively low, right? Because the sodium is being pumped out all the time. Now, in addition, the membrane potential is also kept relatively low, right? Minus 70, that's a familiar membrane potential to us, right? So we have two things favoring the movement of sodium across this cell. We have electrical attraction to the negative inside, and we have diffusion gradient as sodium is being pumped out. So we have two forces that are moving sodium across this membrane, right? So this movement of sodium, which is highly active, I mean, we looked at the numbers earlier, is the primary draw for water reabsorption too, right? So we're absorbing a ton of sodium through this system, and that's going to get us a ton of water too, okay? Just like we would expect water to follow salt. All right, I think that's all those pieces. All right. Okay, so that's the active transporters. The sodium-potassium pumps are driving the show. Now, because the sodium gradient is kept low, in other words, the sodium concentration inside the cell is kept low, it means that we have the potential to use sodium as secondary active transport, right? Sodium's going to fall down its concentration gradient. We want it to take other things with it, either down their concentration gradient or up. So we have a couple of key transporters here that use the sodium-potassium pumps here to drive motion. One you're going to hear about all throughout this year, believe it or not, is the SGLT transporter, okay? This is a sodium-glucose cotransporter. Sodium is falling down its concentration gradient, okay? And technically glucose is falling down its as well, okay? Because remember, inside the cell, glucose is typically relatively low, all right? Now, why do you keep hearing about these this year? There's a new class of medication that affects this transporter called an SGLT2 inhibitor. And it has become a very important antihypertensive in patients who have type 2 diabetes, right? So you're going to keep hearing about it. But the SGLT receptor is this one, sodium glucose cotransporter. So technically it's powered by the sodium-potassium pump keeping the sodium concentration in the cell low. Okay, so that's SGLT. Another is sodium amino acid cotransporters. Okay, so here sodium's falling down its gradient and pulling in amino acids along with it. Both of those, remember where we are, right? We're in early part of proximal convoluted tubule. We've got a lot of filtered glucose, a lot of filtered amino acids, both of which we want to 100% recover, right? Because they're useful molecules. So these two transporters are doing that. They're helping us recover glucose and recover amino acids from filtration, okay? Now, next up is the sodium hydrogen exchanger, the NHE, all right? Here, sodium is falling down its concentration gradient. Hydrogen ion is being pushed up its concentration gradient. Now, if you look, that arrow is in a different direction, right? Because the hydrogen ion is going out into the lumen. So this is technically secretion, not reabsorption, okay? So our secretion of hydrogen ions actually begins in the proximal convoluted tubule. The bulk of sodium-hydrogen exchange, though, is going to happen in the distal convoluted tubule where it's carefully controlled, okay? But it is present here. And then the last process, which wasn't in the picture, but it is important. Okay, so The glomerular filtration membrane, while it is most of the time works perfectly, like everything in the body, nothing ever works perfectly 100% of the time. So there is always a small amount of protein which does get through the filtration membrane. Okay? So how does that protein get reabsorbed? Protein, great big huge thing. It's not like you can just run it through a transporter, right? Because it is the size of a transporter. So there is a small amount of pinocytosis that happens, and I have just realized in my hurrying to put the circle up, I put the circle on the wrong side. Sorry, the circle should be over here. Oops, sorry about that. Pinocytosis, right? So you've got a little bit of protein in the lumen, right? A little bit of protein managed to squeeze through the filtration membrane. This membrane is attractive to protein, so protein binds, pinocytosis pulls it in, the protein is digested, and the amino acids are then reabsorbed. Okay, so I apologize. I was so proud of myself too that I had made a whole circle in just a few minutes, but it should be over there. So please move that in your slide. All right, so, and that's just sort of like it's a backup system. It's not happening a lot, and frankly, it doesn't happen very well. So if you have a lot of protein in your urine, guess what? The pinocytosis system isn't going to cover it. You're just going to spill protein in your urine, right? And we'll call that proteinuria, and we'll work you up for it if you're a patient. Okay. So our secondary active transport system, and then our ATP, or our pinocytosis, which is an ATP using system. All right. This graph should look familiar because we actually talked about it in our first week together when we talked about how enzymes have particular characteristics. One of those is saturability, right? So if you have, and now I can show you why for glucose. You don't have an infinite number of those, right? You don't have an infinite number of SGLT transporters. I didn't mention the GLUT transporter. The GLUT transporter it's a passive carrier transporter that glucose is moving down its concentration gradient towards the blood. Now, why down? Because this is essentially pumping glucose into the cell, right? The SGLT2, because remember, sodium is falling, so that's moving glucose in relatively quickly. It is generally true that glucose doesn't live in cells for very long, right? Because it gets utilized. But here, because of the speed of it, glucose concentration in the cell does increase a bit, enough to have a concentration gradient from cell back into interstitial fluid. Okay, so that's the GLUT, passive carrier-mediated transport, driven by the concentration gradient created by SGLT. Yeah.
So would that be by diffusion then?
Yeah.
Okay.
This is active transport, or this is co-transport.
Co-transport, then
This is diffusion. Yes. Good. Okay, so with those two, if both the GLUT and the SGLT are both working as fast as they can, well, if you add more glucose, you don't get any more reabsorption, right? Because they're all busy. So that's the point at which we start to see urine, or we start to see glucose appear in the urine, right? So, in other words, as plasma glucose goes up on the X, we have filtered glucose on the Y. Okay, generally, in the normal range, the higher our glucose level, the higher the reabsorption, right? But when we get to the saturation point, the transport maximum, as we have more glucose, we can't reabsorb anymore, and we start to see it be excreted. All right, so that's sort of the how we use glucose in the urine to diagnose diabetes, because if you have glucose in your urine, it means your sugar is at least 300, right? Double normal. So it's a quick and dirty test. All right. Now, the most famous of the saturability is glucose. But this occurs all throughout the reabsorption pathway. Anything that has a transporter is going to have saturability, okay? Because there's only so many pieces that can work so fast. So like this happens for phosphate, this happens for sulfates, This happens for amino acids, urate, lactate, plasma proteins. Now, the reason I have PKU here is because by now we will have talked about the metabolic diseases, right? So in PKU, we have extra high amounts of phenylalanine, right? Well, usually phenylalanine is completely reabsorbed, just like glucose. But in patients with PKU, they have so much phenylalanine that we start to see phenylalanine appear in the urine, right? So it's the same pattern. Yes?
Does the body ever create new or like more like sodium glucose co-transporters?
Like to adapt,
Right.
like to upregulate. There doesn't appear to be a successful upregulation, which is interesting. But it is probably related to how the glucose gets high in the first place. But no, you would think it would, but it doesn't. All right, so reabsorption limits saturability, and remember phenylalanine will play the same role as glucose will. All right, so we can't forget about water. You know, solutes are easy to follow around because they have molecular machinery that moves them. Water doesn't. Water is passively moved around by osmotic pressure. Okay, so what that means is we have to look at the sodium moving and then determine where the water is going, right? So as the sodium is reabsorbed, let's go back here for a minute, right? So this sodium-potassium pump is going to have a tendency to reabsorb sodium in large amounts, all right? Well, as the sodium moves, the water's going to follow, okay? Not through the same transporter. Water has their own channel called aquaporins. Aquaporins are found in the vast majority of cells. In the kidney, we have two spots where they don't exist. At least they don't exist all the time, and I'll point those out. But typically, cells are permeable to water. So we move the salt, the water follows. Now, it is also true because of the paracellular pathway, right? Remember, cells have small gaps between them that small molecules can get through. That means that just like water follows salt, salt also follows water when it can. In other words, if it's going through a gap where the water goes, some salt's going to come along with, right? So we can move the salt ion and the water will follow, or we can move the water and the salt will go with it. Do you see? Okay, so we have both things. So the sodium reabsorption drives water reabsorption. The water reabsorption does in fact drive some reabsorption of solute. So we get a little of both. Okay. Now, considering the volume, we saw in that first table how significant reabsorption is, right? So we're getting both effects in those big numbers. We're holding on to the water and we're holding on to the solute. All right, so if we look a little more closely at that passive reabsorption, right? So we absorb sodium, okay? That's going to trigger absorption of water, right? Absorption of water is going to trigger an increase in luminal chloride concentration. Why? Because you took the water away, right? So remember, one of the ways to increase concentration is to remove water. So that's what we've done here. We've taken water away. That's increased the concentration of both urea and chloride. Urea is passively reabsorbed along with some water. Chloride is passively reabsorbed along with some water. Now also, sodium reabsorption tends to leave the lumen negative. Okay, so let's go back and look at this. If we have a bunch of positive ions that are moving across here, well, it's going to tend to leave the filtrate slightly negative, right? That slight negativity repels the chloride, right, and therefore drives additional chloride reabsorption. So we have absorption of sodium, water, and the things that it triggers. So both chloride and urea are traveling down the concentration gradient, and then chloride is also being driven by the luminal, by the increased chloride concentration in the lumen too. right? So this increased concentration is going to tend to push chloride into the body. So we get both sodium and chloride moving across the cells, and therefore water follows, right?